Okay, hi everyone, welcome to lecture 8 of MD6028. Again, we're into the kind of pathogenesis portion of the module now, where we're talking about the relationship between viruses and human disease. So we're really looking at the different ways viruses can cause human disease, looking at the different types of disease we see. And this brings us on to today, where we're talking specifically about viral oncogenesis. So this is a term we've touched on previously, and we've discussed how it defines the ability of viruses to cause cancer. Essentially, that's what we mean by oncogenesis. Onco meaning cancer, genesis meaning the origin of something. So we've touched on this idea that some viruses, as a consequence of how they infect, they can induce cellular changes that lead to the formation of cancers. And when we're talking about the formation of cancers at a cell biology level, what we're talking about is the ability of cells to hyperproliferate and therefore produce a mass of cells, which essentially is what a tumor is, right? So again, viruses are doing something, or some viruses are doing something that confers this ability to hyperproliferate onto the infected cells. And what we want to do today is think about what this is. So what is the process by which an infecting virus can confer this ability to hyperproliferate on the infected cell? It's not a straightforward question to answer, in the sense that there's not a single mechanism by which all the different oncogenic viruses, and oncogenic virus means a virus that does this, that causes oncogenesis. Again, there's not a single mechanism that all these oncogenic viruses use. So what we want to do is look at the different oncogenic viruses that exist and look at the different mechanisms by which these different viruses cause oncogenesis. Now, we've split them into two large groups because largely we see these two significant distinctions. We see the RNA viruses and the DNA viruses, and the way that the oncogenic viruses within these groups cause cancer, so cause this process of hyperproliferation, is very, very different. So the oncogenic RNA viruses cause this hyperproliferation in a different manner to the oncogenic DNA viruses. So we've grouped them in this way. Again, you should understand these distinctions because we've talked about the phenotypic classification of viruses. We've talked about how we can split RNA viruses from DNA viruses because they have this different genome, an RNA genome versus a DNA genome. And we've talked about how there are some real observable differences and consequences to the difference in genome. And this is another example of that. So again, we're seeing that there's this significant difference between the RNA viruses and DNA viruses, and that's why we're splitting them up for this lecture essentially. Okay, so first we're going to talk a little bit more about oncogenic viruses more generally, because we want to make sure we've established all the necessary definitions. We need to make sure we understand all the fundamental concepts as we go into the lecture. Okay, so a straightforward definition to start. We need to understand what we mean by cancer. As I'm sure we all know anyway, a cancer is essentially a mass of cells that have resulted as a consequence of hyperproliferation of individual cells. So individual cells lose the ability to regulate their cell cycle, which is something we'll talk about in detail in a little bit. And that essentially means they can keep replicating and replicating and replicating. And this is hyperproliferation. And it means you get a large number of cells produced, because whilst replication of eukaryotic cells is normally very tightly regulated, when this regulation is lost, so when there's hyperproliferation, it can really quickly lead to the formation of a tumor, which is again oncogenesis, because a tumor is just a mass of replicated cells. So really, if we think about the phenotype of what we're seeing here, so the outward characteristics, we're seeing uncontrolled growth. So that's the main phenotype, isn't it? That's the main observable characteristic. These cells have been modified by the infecting virus in such a way that they are replicating and they are hyperproliferating, and this is what causes cancer formation. We use a specific term for the change that undergoes a cell when it moves from being a healthy cell with a normal replicative rate to being a cell that has an uncontrolled replication, so it is hyperproliferating, and we use the term transformation. So we say the cell has been transformed. And again, an untransformed cell is just a normal cell that replicates in a very controlled manner, usually something like once every 24 hours. However, if that cell can become transformed as a result of this transformation, the cell hyperproliferates, so it replicates much quicker. It just continues to divide and divide and divide, and obviously we can see how this can result in the formation of a tumor. So again, this process is referred to as transformation. Transformation can happen naturally via mutations and things like that, but it can also happen as a result of viral infection, which is what we're talking about today. Again, when we're thinking about what this process called transformation means, in the context of cancer, we're thinking of it as a prerequisite to cancer formation. So the cells become transformed, then they have this hyperproliferative state because they're replicating, and this can lead to the formation of cancer. So this is a kind of prerequisite for the formation of cancer. So again, when we're thinking about these viruses, so we're thinking about the process of viral oncogenesis, we're thinking about viruses being able to induce this process called transformation. So it's a cellular process that is a precursor to the later stages that ultimately lead to oncogenesis. So transformation enables proliferation, so this hyperproliferation of the cells, which understandably leads to the formation of a tumor. These are the key fundamental bits of information we need. So transformation is the early stage, and this leads to cancer formation. We've referred to the cell cycle a few times. This is what the cell cycle actually looks like. Again, you've probably discussed this at some point in cell biology lectures. If not, I'd encourage you to do a little bit of reading on what the cell cycle is. For the context of this lecture, what we really need to know is that it's a process through which dividing cells go, and this process is again tightly regulated. So cells will progress through this in a clockwise manner, so from M phase, where mitosis occurs, to G1 phase. So G1 phase is when the cell is growing. S phase to when the genetic material is being replicated essentially, then G2 phase, which is the second growth phase, and then again M phase, which is the mitotic phase, so when the cell actually physically divides, and these subsequently produced cells will then go through the cell cycle themselves. This is a cycle, a continual process. Crucially, however, it's a very well-controlled process, and generally speaking, we can think of the cycle as taking around 24 hours in a healthy dividing cell. However, if transformation takes place, this cell cycle is progressed through very rapidly, and as a consequence of this, we see hyperproliferation, because that's what hyperproliferation is. There's consequences downstream of hyperproliferation. One, as we've said, tumor formation, but the other is that the cell becomes more and more damaged. If we see a cell that has been hyperproliferating, so if we look at, tumor cells, we see that genetically they're a mess. And the reason they're a mess, the reason why they're kind of all over the place at a molecular level, is because the closely regulated nature of the cell cycle is there for a purpose. It's there to make sure that the cell is prepared before it progresses to the next stage. Okay, so each of the cell cycle stages serves a purpose. There's certain molecular things that should be going on, and if these molecular things, these processes aren't complete, then the cell isn't ready to progress to the next stage. If progression happens anyway, so the cell cycle checks aren't in place, which is again what happens in hyperproliferation, then the cell becomes kind of more and more damaged, and it accumulates more and more DNA damage crucially, so genetically, again, it's just kind of all over the place because it doesn't have the time to replicate its genome properly. So again, it just accumulates more and more damage. And we need to reinforce this point that in a healthy cell, the cell cycle is really closely regulated, and these are the two regulated checkpoints between G1 phase and S phase, and between G2 phase and M phase. And again, these are checkpoints because at these points there are molecular mechanisms which determine whether or not the cell is ready to progress to the next stage of the cell cycle. And if these molecular mechanisms detect that the cell is ready, then it is able to progress into the next stage. If they determine that the cell is not ready, then this progression can't occur. If the cell is transformed, this control is completely lost and the cell progresses to the next stage whether or not it is ready at a molecular level. And again, this results in the accumulation of damage. If you're looking at the cell biology literature relating to cancer cell biology, you'll see a massive focus on these two cellular checkpoints, G2M and G1S. And further, what you'll see is a huge number of proteins that are involved in some respects to cancer formation. So they're involved at a molecular level, usually in this process of transformation. These proteins often are involved in the cell cycle. They often have some function in the cell cycle because, again, this cell cycle is what is dysregulated as a precursor to cancer formation, because dysregulation of the cell cycle is what enables hyperproliferation. Okay, so on to oncogenic viruses. So we've talked about these background ideas that we need to understand. We've talked about what cancer is and this idea of transformation, and we've talked about the fact that viruses can be have a role in transformation, so they can induce transformation. What we need to do now is understand exactly what this role can be. And we want to do this with a little bit of context, so we can start thinking about the very first oncogenic virus that was discovered. So it's referred to now as the Roos sarcoma virus. It was discovered by a researcher called Dr. Peyton Roos, who determined that this virus was the causative agent of sarcomas. He essentially was able to isolate the agent, didn't really know what a virus was, but they knew it was an agent, so it was something that was able to do something, and this agent was able to induce tumours in healthy chickens. So he could extract the virus from sarcomas, so tumours in English hens, and then use this agent to induce tumours in otherwise healthy chickens. So again, clearly this is evidence that there was something in the tissue, so in the cancer tissue, that was capable of inducing cancer formation in healthy tissue. And they didn't understand what viruses were then, but they knew there was something doing this, and now we understand that this something was in fact a virus. Specifically, it was a retrovirus. As we'll see in the next few slides, retroviruses in particular are noted for their ability to induce transformation of cells. So retroviruses in particular are known for their ability to induce oncogenesis. There are specific reasons for this. One of the reasons is that, as we know, retroviruses have an ability to modify the host cell genome. And this is something we've talked about in past lectures. If we look back at how retroviruses replicate and express their genome in a cell, we can see they are able to induce changes in the host cell genome. Namely, they are able to introduce, by integration, their own genome into the host cell genome. And again, there are consequences of this that can lead to transformation, which lead to cancer formation downstream. So if you think about all the different retroviruses that we know, and we have many of them listed here. So again, the virus family is Retroviridae, because we're talking about retroviruses. And within this family, we have the genus alpha retrovirus and beta retrovirus. We have them all listed here. And then we have all the species on the right. If we look at the names of these species, it becomes pretty clear that there's a very frequently observed ability for retroviruses to induce cancer formation. And again, what is essentially happening is the retroviruses are inducing cellular transformation, and their ability to do this is tied to their replication cycle, because as we looked at when we were understanding the replication cycle, these viruses, retroviruses, have the ability to induce changes in the host cell genome. They have the ability to integrate their own genome into the host cell genome, and this, as we'll look at today, can have downstream consequences which lead to hyperproliferation, which is occurring as a result of transformation. Again, if we look at the names here, we can see Rous sarcoma virus, which we've talked about, tumour viruses, leukosis viruses, sarcoma viruses, hyperplasia. So we're seeing these terms over and over again. Hopefully it becomes clear when we look at these names that there is apparently a very frequently shared ability for retroviruses to induce transformation, leading to hyperproliferation. Okay, and what we want to do now is look at this in more detail. And as we know, retroviruses are RNA viruses. The family Retroviridae is a family of RNA viruses, and so we can consider the way in which retroviruses cause oncogenesis under the banner of oncogenic RNA viruses, because many retroviruses are oncogenic RNA viruses. So there are two ways in which viruses can broadly induce transformation within a host cell, and both of these ways really relate to oncogenes. Now again, if we break up the term oncogene, we can see what an oncogene is. We can see gene, you know what gene means, onco, cancer. So oncogene is a gene that has the potential to, when expressed, induce transformation, which is this prerequisite stage towards the formation of cancer. And again, the two ways in which viruses can induce transformation revolve around these oncogenes. So infecting viruses can either, one, introduce an oncogene into the cell. So if the virus has an oncogene within its own genome, by infecting a cell, it is essentially introducing this oncogene into the cell. So this is one way in which transformation can occur. The second way is by activating oncogenes that are already in the cell. So there are oncogenes within host cell genomes, and viruses can potentially activate these genes when they infect a cell, and we'll look at some of the specific mechanisms by which this can happen. These oncogenes, generally speaking, as we've said, will have an impact on the cell cycle. So we've said so many proteins that are involved in cancer cell biology are playing a role in processes related to the cell cycle, and this is what we see for oncogenes. So many of these oncogenes are involved in cell cycle regulation or have an impact on cell cycle regulation. And again, this is something we're going to look at some specific examples of. There's a note on terminology that we need to make here. So we can split oncogenes into two categories. We have the V oncogenes and the C oncogenes. So V stands for viral, C stands for cellular. So if an oncogene is introduced into a cell by an infecting virus, so the oncogene is present in the viral genome, and therefore when the virus infects the cell, it brings this oncogene with it. We refer to this oncogene as a V oncogene. It's a viral oncogene. On the other hand, if the oncogene is present in the host cell genome, we refer to it as a C oncogene, so a cellular oncogene. So just a note on terminology, because it's important we distinguish between the two. At the end of the day, oncogenes function in a similar way when expressed. They do things, usually to the cell cycle, that ultimately cause transformation. So they induce changes in the cell, and these changes result in transformation. This process of integration that we've talked about many times now, and we've done so in the context of retroviruses. So we've said retroviruses can integrate their own genome into the host cell genome, can cause transformation through both of the mechanisms we've just defined there. So we said there's these two mechanisms. Viruses can introduce an oncogene into a cell, a V oncogene, or they can activate an oncogene that's already in the cell, so a C oncogene, and both of these processes can occur as a result of integration. And because retroviruses are an integrative virus, they integrate their genome into the host cell genome. This is one of the reasons why we see so many retroviruses are capable of inducing cellular transformation. Again, integration is a really effective way of transforming cells by either of these two mechanisms. So integration can result in the introduction of viral oncogenes into the host cell genome, or integration can result in the activation of cellular oncogenes. So hopefully we can see how these concepts are linked. So it's not just a coincidence that so many retroviruses induce transformation. It's actually a consequence of retroviruses' ability to integrate into the host cell genome, because this process of integration can induce transformation in either of these two ways. Again, either by introducing a viral oncogene into the host cell genome, or by activating cellular oncogenes that are already present in the host cell genome. So this is the link. This is why so many retroviruses are capable of inducing transformation and therefore cancer formation. Now, if we look at these oncogenic retroviruses and we look at the way in which they cause transformation in detail, we see we can distinguish between three groups, and these three groups are quite consistent with what we've just been talking about. So I'll define these three groups, then we'll link them to this idea of V oncogenes and C oncogenes. So firstly, we have the transducing viruses. So these are retroviruses that induce cellular transformation at really high efficiency. So they're very effective at causing cellular transformation. So 100% of the time, really, an infection with these transducing retroviruses will cause cellular transformation. So at a cellular level, it's really almost a certainty that this transformation will occur. There's then non-transducing viruses. So these viruses are oncogenic, but they don't have such a high efficiency of transformation. So there's no guarantee that they'll cause transformation. And we actually see the different non-transducing viruses have different efficiencies, so different percentage rates at which we would expect transformation to occur. Finally, we have the non-transducing viruses that are long latency, so non-transducing long latency viruses, and these are really low efficiency. So these have a fairly weak ability to induce transformation. So they can induce full transformation, but the likelihood of that happening in an infective instance is lower than for the other groups. So we can see these clear distinctions in efficiency, and these clear distinctions in efficiency are linked to how these different retroviruses induce transformation. So the transducing viruses, so these are the viruses that have a really high efficiency when inducing transformation. These carry their own oncogenes, so these carry V oncogenes. Now we refer to them as being cell-derived oncogenes because the V oncogenes originally started out as cellular genes, but they were picked up by retroviruses. So retroviruses essentially acquired these genes from cells, and they then became V oncogenes. So what we're referring to here is a viral oncogene. So when these viruses infect cells, they introduce the oncogene they are carrying, the V oncogene. This oncogene is expressed, and then transformation is induced. The non-transducing viruses, so these who have the kind of varied efficiency of transformation, these don't carry viral oncogenes, and instead they activate cellular oncogenes. And we sometimes refer to the cellular oncogenes as proto-oncogenes because many of them require activation by an infecting virus. So in the host cell, they're a proto-oncogene until they're activated, and we can then refer to them as oncogenes. This process of activation is a cis activation, and that means in the same position, so in the same site. And usually what is happening is the retrovirus integrates next to the proto-oncogene. so physically next to the proto-oncogene in the cell genome, and therefore when the viral genome is being expressed, the proto-oncogene is also being expressed just because it is physically close to the viral genome. And again, all the factors necessary for expression are there. So it's almost like an accident. It's being expressed just because it's next to the retroviral genome that's been integrated. So it's this cis activation. We can compare this to the non-transducing long latency viruses, which have low efficiency of transformation, and say that these viruses also induce cellular oncogenes, so these proto-oncogenes, but the induction is trans activation. So they're activating the expression of genes that are not physically next to the integration site of the retroviral genome. So the difference between these last two groups is whether it's cis activation, so whether the proto-oncogene is next to the site of integration, or whether it's a trans activation, so the site is different. So hopefully we can see these three groups line up well with this idea of viral oncogenes and cellular oncogenes, and hopefully we understand these distinctions. And again, transducing viruses cause transformation by introducing viral oncogenes into the host genome. And non-transducing viruses and non-transducing long latency viruses, rather than introducing viral oncogenes into the host cell, they activate cellular oncogenes. They activate these proto-oncogenes within the host cell genome. And the distinction between these two groups is whether it's cis activation or trans activation, with cis activation meaning at the same site, trans activation meaning at a different site within the genome. So we'll think about some examples now. Firstly, we want to think about Rhesus sarcoma virus, which is an example we touched on earlier because it's the first retrovirus that was discovered. It's a transducing oncogenic virus, so that means it carries an oncogene, it carries a V oncogene, a viral oncogene. Now it's interesting for us to think about where this viral oncogene came from, because this would help us to understand how these viruses acquire the ability to induce transformation, which is obviously something we really care about if we want to understand this process as a whole. Now we've said these viral oncogenes are cell derived, and this is what we see is the case if we look in detail at any one of these oncogenes. For example, the viral oncogene that Rhesus sarcoma virus carries, VSRC, is a version of the cellular oncogene SRC. So we have the cellular SRC gene, and we can see that viral SRC is derived from that. It's just a version of that gene essentially. Specifically what this gene does is it encodes a protein that is a tyrosine kinase, which adds phosphate groups to tyrosine, and by doing so it stimulates uncontrolled mitosis. So again, it induces transformation. So this is completely consistent with what we've talked about, referring to these genes that are involved in transformation. So SRC is clearly having an impact on the cell cycle. It's impacting mitosis, it's impacting cell division. So again, consistent with what we've learned about this process of transformation, oncogenes, when expressed, impact progression through the cell cycle. This is usually what we see. So it's also interesting for us to think about how the virus has obtained this oncogene. So how is it that a cellular oncogene has come to be carried by a retrovirus? We're not quite sure. There's no certainty. Again, it's kind of retrospective, isn't it, trying to figure out how something happened a long time ago. But we can theorize, we can look at the life cycle of these retroviruses, and we can think about what possible events could have led to the retrovirus capturing this oncogene. And to do this, we need to just have a quick recap of what the retroviral genome expression and replication looks like. So I'm sure we remember the retroviral genome is a strand of positive sense single-stranded RNA. This undergoes reverse transcription mediated by reverse transcriptase, and the product of this is double-stranded DNA. This double-stranded DNA is then integrated into the host cell genome. Once integrated into the host cell genome, it can be expressed just like the host cell genome, and the product of that expression is positive sense single-stranded RNA. This positive sense single-stranded RNA can, as we know, be translated into protein, or it can be used as copies of the viral genome, so it can be packaged into new viral particles, and this is essentially the whole process that retroviruses undergo to replicate and express their genome. Now, within the viral genome, so integrated in the host cell genome, there is a stop codon at the end. Okay, so when the genome is being expressed, there is a stop codon to signal the end of the viral genome. So this is where expression should theoretically stop. We say theoretically because it is fairly frequent process by which this stop codon can become mutated and therefore potentially lose function. If this stop codon loses function, then expression can continue past the full length of the viral genome and can include portions of the host cell genome, and these portions therefore get included on the end of the viral genome. Now, if we have a cellular oncogene that is adjacent to the viral genome, which we do sometimes have because we know that this is one mechanism by which integrating viruses can activate proto-oncogenes. Again, they can activate their expression just by virtue of integrating physically near them. So we know that we can have viral integration near to an oncogene. And again, if this happens and there is a loss of function of the stop codon, so the stop codon is mutated, then expression of the viral genome could potentially continue past the end of the viral genome and include some cellular genes, one of which could be a proto-oncogene that is therefore introduced into the viral genome and can then be considered a viral oncogene. So this is one of the theories. This is one of the theories on how cellular oncogenes came to be included in viral genomes. So the idea is a proto-oncogene in the host cell genome is captured into the viral genome as a consequence of the virus integrating next to the proto-oncogene in the host cell genome. Okay, so that should have been clear and hopefully quite interesting. Again, we don't know that's exactly what happened, but it's one theory of how retroviruses captured cellular oncogenes. So it's one theory as to how the retroviruses that do have viral oncogenes originally derived those oncogenes from cellular oncogenes. Okay, there's another classification that we can think about when we're discussing these retroviruses, and it's whether or not they induce transformation in a rapid, intermediate or slow manner, and this is completely consistent with the groupings we looked at earlier between the transducing, non-transducing and non-transducing latent viruses. So we've said transducing retroviruses induce transformation with very high efficiency, and in this new grouping system we'll also say they introduce it rapidly. So transducing retroviruses induce rapid transformation and they do so efficiently. Non-transducing retroviruses have varied ability to induce transformation, so varied efficiency, and we also see there's this kind of intermediate kinetics to transformation. So the rate of transformation, it varies. It's not necessarily rapid, but it's not necessarily as slow as the final group we'll look at. And this final group includes, or this final group is the transducing latent viruses. So again, these are the oncogenic viruses that induce transformation with very low efficiency. And now we can also say that this transformation is not just low efficiency, but it's slow. So these have the ability to induce transformation with low efficiency and transformation that is slow compared to the other groups. And again, the consequence of this slow kinetics is because of the mechanism in which transformation is being induced. The mechanism is through transactivation. So it's not a consequence of the retroviral genome integrating physically next to the proto-oncogene. It's a consequence of the viral genome integrating at varied sites within the genome and inducing proto-oncogenes in a trans manner, so inducing them from different sites. And we're comparing this to the intermediate viruses, so the non-transducing viruses that are non-latent, so this middle group that have intermediate kinetics, and their varied ability to induce transformation and their varied kinetics, so this intermediate kinetics is a consequence of their ability to activate cellular proto-oncogenes by integrating physically next to them. So it's this cis activation that we talked about. So again, 3 groups here that are completely consistent with the 3 groups we talked about earlier. So there's this complete overlap between transducing viruses and rapid viruses, and non-transducing viruses and intermediate viruses, and slow kinetic viruses and non-transducing latent viruses. They're the same groups, we're just looking at them from a different definition. And again, transducing are rapid, non-transducing are intermediate, and latent are slow. Okay, so those are the oncogenic RNA viruses. What we'll think about now are the oncogenic DNA viruses. So as I'm sure we know, oncogenic DNA viruses are really significant. There's a number of DNA viruses that cause significant human cancer. If you think about HPV, that's the most obvious example that I'm sure we're all aware of. So this is human papillomavirus, so member of the Papillomaviridae family, has a very clear and well-defined ability to cause human cancer, so cervical cancers as well as anal cancers and oropharyngeal cancers. So it's really important we understand exactly what's going on here. So the process is somewhat different to RNA viruses, but there's also these similarities. So the overall similarity is that there's an impact on oncogenes. There's this ability to induce cellular transformation through oncogenes. Now the specific oncogenes that are being impacted can be different, but that's the principle. We're seeing again transformation as a consequence of oncogenes disrupting normal cell cycle progression. You'll see this term T antigen crop up again, and T antigens are observed in many DNA viruses that induce cancers. So T antigen is kind of used as a bit of a general term to refer to oncogenes that are carried by these DNA viruses. You won't always see it used, but it's for ease of use, it's easy to refer to them here. So we know we're talking about these DNA virus oncogenes that are again inducing transformation within the host cells. And the process is similar in the sense that these oncogenes are inducing dysregulation of the cell cycle, and in doing so they are inducing cellular transformation. So to understand exactly how DNA viruses can induce transformation, and also the specific mechanisms by which the RNA viruses are inducing transformation, we need to look in more detail at these oncogenes. So we've said RNA and DNA viruses induce transformation by either introducing oncogenes into the cell for DNA viruses or some RNA viruses, or for activating cellular oncogenes for some other RNA viruses. but we need to know exactly what these oncogenes are. So how are these oncogenes inducing transformation? Now, we've said they're impacting cell cycle progression, and that's absolutely true, but we need to know a little bit more than that, don't we? So we're going to look at it in a little bit more detail now, and we're going to try and link up this gap between the oncogene being present in the cell, whether it's introduced by the virus or it's already there and it's been activated by the virus. So we're going to link this to the actual process of cell cycle dysregulation, and then we can see hopefully clearly how it leads to cancer formation. So once we're at the point of cell cycle dysregulation, there is hyperproliferation, and again this leads to cancer formation. So the only missing link we really need to understand here is how these oncogenic proteins, these oncogenes, induce cell cycle dysregulation. So we can say there's two broad ways in which these oncogenes can actually cause cell cycle dysregulation. The first is by impacting cellular signaling pathways, and we'll look at what this means in a couple of slides. And the other is by actually dysregulating the cell cycle through the particular checkpoints. So we've said the cell cycle is this cycle through which cells progress, and there are specific checkpoints, and these checkpoints determine at a molecular level whether the cell is ready to progress to the next stage of the cell cycle. And what some oncogenes will do is they will compromise these checkpoints, and in doing so they'll dysregulate the entire cell cycle, because these checkpoints are the points at which control is normally exerted by cellular mechanisms. Okay, so firstly, oncogenes impacting cell signaling pathways. Now it's important we realize that this cell cycle is occurring in the context of wider processes. So there's extracellular signals that are signaling to cells whether or not progression through the cell cycle should occur. And these signals are one of the factors that determine whether cell cycle progression will occur. So there are these signals which essentially tell a cell, yes, you can divide now, or no, you should not divide now. And some oncogenes can essentially manipulate these cell signaling pathways, and as I'm sure we can guess, they manipulate these in such a way as to give a constitutive yes, you are okay to replicate now signal. So the cell is receiving the signal that it is able to replicate, that it is able to divide, and therefore this hyperproliferation occurs. Now we have an example here, which is SRC. SRC is an oncogene that we've looked at before. It exists in its cellular form, so in its proto-oncogenic form, cellular SRC, and it also exists in a viral form, so VSRC. And this viral form is a form of SRC that induces constitutive activation of these pro-proliferative signaling pathways. So cellular SRC, it induces pro-proliferation as well, so it's pro-proliferative. However, the viral version of SRC is constitutively active. So it's slightly different to cellular SRC because it is not a protein that can be switched off. It's not a protein that's controlled. So this is how it's different to cellular SRC. It's again a constitutively active version. So when the virus infects the cell, it introduces viral SRC into the cell, and viral SRC signals for the cell to proliferate. So it signals for the cell to progress through the cell cycle, and in doing so, it causes transformation. And again, we can see this here. So viral SRC encodes a constitutively active form of cellular SRC. This form is able to induce proliferation because it's a pro-proliferative protein. It's inducing pro-proliferative pathways that we have defined here, and these lead to cellular division, so it's progression through the cell cycle. On a side note, we can see how cellular SRC can also function as an oncogene, because we can have normal cellular SRC, which can function as a pro-proliferative protein when it's induced within the cell. But then if we can imagine a retrovirus integrating nearby SRC and causing huge amounts of SRC to be produced, then this can have a pro-proliferative effect. So hopefully we can see how this cis activation of cellular proto-oncogenes can potentially work. It's mechanisms like that. And that's not quite what's happening here. What's happening here is a viral version of the gene is being introduced, but hopefully we can see the distinction between those two types of activation. And if we want another example that actually does occur of how cellular oncogenes can be activated, we can look at MYC. So MYC is a cellular proto-oncogene. And when it is expressed, it promotes cell cycle progression. So therefore, it is a pro-proliferative protein. And if it is overexpressed, this could be a move towards transformation. So we have Rhus-associated viruses 1 and 2. It's not the same as Rhus sarcoma virus. Still a retrovirus. They are known to induce B-cell tumours, and the way they do this is, again, cis activation of the cellular proto-oncogene. So they integrate near to this MYC protein, or this MYC gene, sorry, and in doing so they cause cis activation of this gene, so you get this massive expression of MYC, and this huge increase in MYC levels leads to pro-proliferation, so it induces transformation. The other strategy we introduced is the idea that these oncogenes can just directly target the cell cycle itself. So rather than targeting upstream signaling, they can directly impact the cell cycle. And this is what's occurring in the case of T antigens. So T antigens being oncogenes that are carried by some DNA viruses capable of inducing transformation. So these T antigens can bind to retinoblastoma. So retinoblastoma is a protein that controls cell cycle progression. So it specifically prevents progression from G1 phase to S phase. So it maintains the integrity of the G1S checkpoint. So it's controlling progression through this checkpoint. Now, T antigens can bind to retinoblastoma, and in doing so they can prevent retinoblastoma from inhibiting progression through this checkpoint, and therefore the T antigen is essentially enabling the cell to progress through this checkpoint unchecked. So it's dysregulating cell cycle progression. So some DNA viruses, by introducing T antigens into the cell, are compromising this G1S phase checkpoint, and in doing so are pushing the cell towards transformation. As a side note, you'll see this term tumor suppressor gene sometimes used, or tumor suppressor protein. Usually this refers to a protein that is protecting the integrity of the cell cycle. So it's maintaining the cell cycle, because by doing this, by protecting the cell cycle and controlling progression, the protein is preventing tumours from forming, and the formation of tumours often requires the inactivation of these tumour suppressor proteins, and that's what we're seeing here. Retinoblastoma controls progression from G1 to S phase, it prevents progression through, and therefore for transformation to occur, it's necessary for these DNA viruses to inactivate retinoblastoma using T antigen. So we want to look at this process in a little bit more detail. So we want to understand the specifics, because it's a really important example of how DNA viruses can induce transformation. So let's think about what's happening in a normal cell again with this retinoblastoma. So as we've said, retinoblastoma is controlling progression from G1 phase to S phase. So in a situation where progression should occur, so there is pro-replicative signaling that is triggering the cell to progress through the cell cycle, retinoblastoma is phosphorylated, and when it's phosphorylated, retinoblastoma is not able to bind to a protein called E2F. Okay, E2F is a transcription factor that promotes progression from G1 phase to S phase, because E2F essentially induces the expression of genes that are required for S phase. So when retinoblastoma is phosphorylated, pro-replicative signaling causes phosphorylation, remember. So it's pro-replicative signaling, retinoblastoma is phosphorylated, and then when it's phosphorylated, it dissociates from E2F, and this means E2F can induce the expression of S phase genes, and this enables the progression from G1 phase to S phase. We have this demonstrated clearly here. So this is the process I've just described. So the normal pro-replicative state of a cell. And again, remember, a cell is not always pro-replicative. This is what's happening when it is given the signal to replicate. So normally, retinoblastoma is preventing a progression through the cell cycle by binding to E2F, but this is what's happening when the cell receives the signal to replicate. So there's pro-replicative signaling. This causes the phosphorylation of retinoblastoma. Retinoblastoma then dissociates from E2F. E2F is then able to induce genes that promote S phase, and this mediates the progression from G1 phase to S phase. Okay, so all this is in response to pro-replicative signaling. Now what happens during adenovirus infection? So in adenovirus infection, we have the instance where we looked at in the first slide, where retinoblastoma is being impacted by a T antigen, because adenoviruses are, some adenoviruses are capable of inducing cellular transformation, because some adenoviruses are oncogenic viruses, and again, these are DNA viruses. We know adenoviruses are DNA viruses. So the T antigen essentially binds directly to retinoblastoma, and by binding directly to retinoblastoma, the T antigen is preventing retinoblastoma from inhibiting E2F, and E2F can therefore induce S phase genes and mediate this transition from G1 phase to S phase. Now this is happening without pro-replicative signaling. So normally this activation of E2F only happens when the cell receives a pro-replicative signaling. However, adenovirus is bypassing this. It's preventing retinoblastoma from inhibiting E2F, even when there's no pro-replicative signaling, and this is how adenovirus is inducing this pro-replicative phenotype without pro-replicative signaling. So the adenovirus is causing transformation by inactivating retinoblastoma. And here we have some other oncoproteins. So the proteins expressed through oncogenes, so the proteins that oncogenes encode, that have a function of disrupting retinoblastoma. So retinoblastoma is really central again to this G1S checkpoint. So it's unsurprising that a few different oncoproteins carried by different DNA viruses are capable of inducing transformation by targeting retinoblastoma. Another important tumour suppressor protein is p53, so you sometimes see it referred to as the guardian of the genome, because in a huge number of tumours we see p53 is non-functional. It's been overcome because when p53 is functional, it is very effective at preventing transformation. So often when transformation does occur, it's because p53 has been compromised, or it's just not functioning or something like that. Normally it detects in response to DNA damage or inappropriate DNA synthesis, and it essentially initiates cell death. So if p53 detects that the cell has progressed through the cell cycle inappropriately, so it's gone prematurely into S phase, it detects the consequent DNA damage and kind of aborts the cell cycle replication in such an extreme way that it causes cell death, because this is a preferential outcome to hyperproliferation. As we said, a number of viral factors can directly target p53. So some will target p53 for proteasomal degradation. So proteasomal degradation is a cellular process that destroys proteins. Some will bind directly to p53. So there's an adenovirus protein called E1b that will do this. Some will sequester p53. So the LT of SV40 virus is one example. Again, each of these three strategies serves to inactivate, to compromise p53, and in doing so is preventing p53 from causing cell death, and is therefore making it easier for the virus to induce transformation, or at least to induce transformation in such a way that doesn't result in cell death. Okay, right, thanks very much everyone. So that's everything I wanted to talk about today. As always, we'll work through this in the tutorial on Monday, so if you have any questions, feel very welcome to ask them in the tutorial. Thank you.